All right, so we're almost done today, right? So I work at the Bioinformatics Core at University of Illinois, Chicago. So I'm going to talk about one of the projects we've been working on with some collaborators at University of Chicago, which are easily confused. So I want to start off, do we have a laser pointer up here? Oh, here we go. I'm blind, sorry. Our collaborators at University of Chicago have produced some very nice data that I get to analyze. And the rest of my bioinformatics team, my director and my coworkers at UIC. And then I know that most of us here are probably bioinformatics experts, but if anybody is looking for bioinformatics help, our course facility serves not just UIC, but we have projects with other Chicago area research institutions and a few universities across the country. So we're open to anybody. So the project of interest today, our collaborators are studying B-cell development, and a lot is known about what happens as B-cells develop from progenitor stem cells through a variety of intermediate stages before they become finally mature B-cells as part of the immune system. These changes are characterized by differences in cell surface markers that come and go over the course of different cell states, characterized by changes in the antibody gene content, VDJ recombination, and also characterized by certain epigenetic factors like the presence of key transcription factors that control the differentiation out of certain cell states. Their goal was to get a kind of higher level picture of chromatin state changes across B-cell development, something a little bit more broad than specific transcription factors. And so for this, they were turning to ATAC-seq, which I think we may have heard a little bit about already, and we'll hear about more tomorrow. But it's an experiment analogous to DNA-seq. You can measure open chromatin. You can also measure the converse of that, nucleosome positioning, if you do paired-end sequencing. So it's a very nice way of measuring the activity and changes in the activity of cis-regulatory regions across a number of different cell states. And then you can, of course, relate this to other epigenetic factors, associations with gene expression changes. You can do motif analysis that we've heard a lot about today. But our goal with this data set first was just to define what set of patterns we observed for changes in open chromatin. So the basic data sets we were looking at here were 8 different cell states. We had actually 2 subtypes of progenitor cells from a publicly available data set that was published earlier this year. And the basic workflow for this, we start with standard peak calling routines to obtain the likely open chromatin regions in each site. But since individual peak calls are always subject to a thresholding problem, I have to choose some threshold at which I call a peak. And depending on where I set that threshold, I may or may not call a peak for certain data sets, or I will call it for others. So we do a strategy where we merge the peaks across all, here 3 are shown in this cartoon, but all 8 data sets, and then go back and count the raw reads, the raw read alignments in each of these peaks from each data set. And here you can see more of a continuous distribution of enrichments, because I'll see a few, maybe a few reads in this peak where there was no peak called for that sample. But once I have these read calls, I can do differential expression with a program like EdgeR. It's written for RNA-seq, but it does differential expression, differential enrichment for count data. And I can compute p-values for changes across all the conditions, so an omnibus test, and I can look at various pairwise p-values and fold changes across my different peaks, or across my different conditions. From here, so we can filter for significantly differentially enriched peaks using this omnibus p-value, and I constructed feature vectors in this case from the product of the log p-value and the log fold enrichment, rather than looking at just the normalized enrichment levels themselves. The idea being we want to capture as much of the significant variability as we can. And then this kind of led into the main challenge for the project was how do I cluster all the huge number of regions that I have into something that's digestible and interpretable in light of other data sets. So to give you an idea of the scope here, we ended up with about 110,000 peak calls, regions of interest after filtering for significance. And the our common approach that's very nice for these kind of data sets where we want to use pattern and discovery is to use hierarchical clustering, plot the data on a heat map, and just kind of study the heat map for a while and see what sort of patterns appear. This was not particularly feasible for this many regions. Hierarchical clustering does not scale particularly well either in computational expense or in memory requirements. I think I couldn't even compute the distance matrix in R. R would just crash when I tried to do it with this many regions. So I turned to k-means in this case, which is a nice lightweight algorithm, but as we all know, k-means is sensitive to your choice of initial conditions, and you have to choose how many clusters there are, which is kind of the point. If you want to know how many clusters there are, it doesn't help to have to choose a priori. So the strategy was to repeat k-means over a variety of values of k, and then to run it multiple times at each choice. And then over your multiple runs, we can compute this cumulative density function of a consensus value index to demonstrate what the right value of k is. And what this means more specifically, so this idea comes from this paper that was published last year, not my paper, but it was a very nice review of some of the pitfalls you can easily fall into, in this case with consensus clustering. They showed for some examples in simulated data where they simulated data from a patently unimodal data set, so something with no clusters, but then they could use consensus clustering heat maps to convince themselves that there were in fact 2 clusters, or 3 clusters, or as many clusters as they chose to look for, just based on accidental chance random clusterings from the data sets. Oh, this is the fun of different computers. Wow. Well, this isn't useful at all, is it? So there's supposed to be, let me just skip to this. All right. So the idea was you plot across your set of co-clustering, repeated clusterings, you're looking at the fraction of times that a pair of samples is co-clustered, is clustered in the same plot. So this is actually my data, but pretend it's simulated data for theirs. What they showed is you adjust a threshold for co-clustering. So the fraction of times that 2 items would have to belong to the same cluster to consider them co-clustered in your consensus clustering. You can look at what fraction of samples end up co-clustered as you adjust the threshold. And the idea is, when you plot this for different clustering levels, so here are my different colors or different k values that I used in clustering, that you should see a very flat curve for correct values of k. And for incorrect values of k, you'll see a curve that gradually drifts up in a non-flat manner. Intuitively, what this means is that every time you repeated your clustering, you got almost the same answer. And so the number of co-clustered pairs was insensitive to this consensus value index, this threshold on clustering. This figure was supposed to be a nice demonstration of the simulated data that they published with the paper that showed how this worked for k equals 1, 2, 3, and 4 for simulated data. You'll have to imagine it, or you can look up the paper if you're curious, but you can take my word for it for now. But then, okay, for now for our data, which we've been looking at for a while, what we saw kind of interestingly was that there wasn't a single value of k that gave us this flat curve. We actually had a lot of options. So k equals 2, 3, 4, 6, 7, 8, 9, 10, 11 are all perfectly flat or almost perfectly flat curves, so totally insensitive. And so basically what this meant is that I had a number of options for how many clusters I could choose and still get a robust clustering of the data even over repeated runs of k-means. 5 and 14 are kind of highlighted as little parentheticals because they were close to flat, but not quite. So it was kind of an interesting result that we had a lot of options for clustering. Moving forward for this, I chose k equals 11, figuring that it would give us the greatest granularity for differentiating different types of patterns when we're thinking about associating these clusters with other types of data downstream. So just to give you a quick illustration of what a couple of these clusters look like. So these are two clusters, cluster 8 and cluster 10. The numbers don't mean anything. But I'm plotting along the top the open chromatin average enrichment for all the peaks that belong to that cluster for each of our 8 samples. The bottom is a nucleosome differential, so I said you can use ATAC-seq for nucleosome positioning information. It's basically the converse of open chromatin. If there is open chromatin, then there's a lack of a nucleosome, and a negative value here means a lack of a nucleosome. But cluster 10, when you look at this, you can get a very easy biological interpretation. We have a lot of enrichment in the pre-Pro-B stage, a little bit of enrichment in some of the progenitor stages, and then pretty much very low or no enrichment as in the later B cell developmental stages. And you can contrast this to instances like cluster 8 on the left, where you can see more of a gradient across different stages. I have several stages with high enrichment, a few stages with medium enrichment, and a few stages with low enrichment. And I think one point to make here is that all of these conditions show enrichment for this set of clusters. And so if you looked at peak calling, if you compared peak calls, you would probably have peak calls for all 8 samples, or all 8 conditions in this cluster. But only by looking at the differential enrichment can you see this actual level of, this actual gradient, these actual differences in the enrichment levels. It also worked out pretty nicely that the 11 clusters we had split up the 110,000 peaks reasonably evenly. Each had around 6,000 to 16,000 regions. Okay. So that's the basic summary of where we are right now. This is a work in progress. My collaborators are collecting RNA-seq data as well as ATAC-seq data for a number of other mutated B-cell developmental conditions. But I think we're excited about using this data set as kind of a core library to compare to other epigenetic factors, to compare to transcriptional changes, and something that we can use moving forward for more studies. And if you're interested in hearing any more, I have a poster upstairs as well. I'm poster 9. All right.
Thank you very much. So there is time also for questions while